Okay, so good afternoon, guys. Welcome to the second lecture of the host pathogen interaction. Okay, and I am Rishabh, and in this lecture, we'll be discussing about lymphoid organs and how the cells are actually involved in the immune response. Okay, so let's start. So the starting with the primary lymphoid organs. So the lymphoid organs, as we can see, so the lymphoid organs are the ones that actually help in developing as well as maturing the immune cells. Okay, so these are basically talking about the lymphoid organs. They help in producing, maturing, and making the immune cells active for function. Okay, so this is the basic role of all the lymphoid organs that we see. Okay, now the lymphoid organs are divided into two categories. First is the primary lymphoid organs, and the second is the secondary lymphoid organs. The primary lymphoid organs are basically helps in the production and maturation, the maturation of the lymphocytes. Okay, and the secondary lymphoid organs, on the other hand, helps in differentiating different lymphocytes and the immune cells to fight against the infections. Okay, so this is the major role of the primary and the secondary lymphoid organs. So the secondary lymphoid organs basically helps the immune cells to get activated and get actually like specific against the against the pathogens. Okay, so talking about the primary lymphoid organs, we have two primary lymphoid organs. First is the bone marrow, and the second is the thymus. Okay, so bone marrow is basically present in the core of the bone, and it has two parts. First is the red marrow and the yellow marrow. Okay, so the red marrow is part of the spongy bone, and it helps in production of different immune cells and the blood cells. Okay, and the yellow marrow is basically a component that is filled with fat. So we started with the bone marrow. Bone marrow has main two components. First is the red marrow and the second is the yellow marrow. Okay. So the red marrow is basically part of the spongy part of the bone, and it contains the cells that form the immune cells and the blood cells. Okay. And the second part is the yellow marrow that basically contains the fat. Okay. Then this red marrow is basically, it contains stem cells or the blood stem cells called as hematopoietic stem cells. Okay, these hematopoietic stem cells give rise to different kind of cells that are involved in blood, having different functions in the blood, for example, RBCs, then platelets, and second, the cells that are important in the immune response. Okay, for example, lymphocytes, then leukocytes. Okay, so all these cells that are involved in the immune response, all these cells are produced by the hematopoietic stem cells that are part of the red marrow of the bone. Okay, then this bone marrow is the basic site of the B cell generation and maturation. Okay, and with age, this yellow marrow that was fat filled, it increases 10% to 50%. Okay, so initially it has a fat deposit of nearly 10%, then it moves to 50% as the age advances. Okay, then this is the case with the humans. This is the case with the humans in which bone marrow is actually helping in maturation and the production of the B cells. But in other organisms, actually bone marrow is not the primary site for B cell generation. Okay, so it is the like for example, in birds, it's bursa of fabricus. That is actually a pouch of the intestine which is located close to the cloaca and helps in B cell maturation in the chicken, or in all the birds. And in cattle and sheep, this is fetal spleen and the Peyer's patches that helps in B cell maturation. And in rabbits, the appendix basically helps in B cell maturation. Okay, so this is how B cell maturation takes place inside in the humans and in different other organisms in which it is actually specialized. Okay, so any doubts for this one, for the bone marrow? Okay, if not, then we'll move on to the next lymphoid organ, that is thymus. Okay, so this is the second primary lymphoid organ. Here, mostly T lymphocytes are matured. So T lymphocyte maturation takes place in the thymus. So where it is located? It is basically located near heart. So this is the primary location of the like thymus, just above the heart, just above the diaphragm. It is located, and this is the primary structure of the thymus. This yellow colored thing that can be seen in the figure. Okay, so it is a bilobed structure, and the at the molecular level, if we see, because it is bilobe, so this is the first lobe that we can see here. So each lobe will have the similar structure. Now this lobe will be actually divided into different lobules. Okay, so one lobe will be divided into different molecular compartment called as lobules. Okay, each lobule will be separated by a connective tissue sheath called as trabecula. Okay, so this trabecula is a connective tissue sheath that separates each lobule. So this is a connective tissue sheath that separates each lobule. Now, this lobule is divided into two components, that is the cortex, the outermost covering, and the innermost region called as the medulla. Okay, so cortex is the outer region of the particular thymus, and medulla is the inner region. Now, the cortex contains all the T lymphocytes that are basically dividing and immature. Okay, so the cortex is the site for maturation of the T lymphocytes. Fine. So as you can see in the image itself, these blue colored cells are the dividing lymphocytes, right? Sorry, dividing thymocytes. Now these thymocytes give rise to the T cells, and these gray colored cells in this particular cortex are called as the nurse cells. These nurse cells help these thymocytes to basically grow, and these cells nurture these thymocytes to help them mature. Okay, now when these thymocytes get matured, these move to the medulla region of the particular thymus, and where they actually interact with the macrophages and the dendritic cells. Okay, so that helps in their further maturation, and then the thymic selection. Okay, so this thymic selection will help the thymus to understand whether the T cells are self reactive or not. Okay, so in the medulla, this thymic selection actually gets completed. Okay, so this thymic selection helps the body to recognize whether the T cells that are matured are self-reactive, sorry, are self-reactive or not. If they are self-reactive, then they will be proceeding to the negative selection, and if not, then these cells will go to the peripheral lymphoid organs through the blood flow. Okay, so this is the role from the medulla. Now, there is another component, that is Hassel's corpuscles, that are present in the medulla region. These are basically when we stain these particular organs in through a particular dyes. So these corpuscles actually stain through eosin. Okay, so that is why these are called as eosinophilic cells that help in the regulation or the development of another type of cells, that is T regulatory cells. Okay, so Treg or the T regulatory cells helps in regulating the function of different T and B cells. Okay, so this was the function of the thymus. So any doubts in this particular lymphoid organ? in thymus, around 80 to 90% of the T cells actually die because of this thymic selection. Okay, the rest 20 to 10 to 20% actually go to the peripheral lymphoid organs for their further differentiation. So self-reactive T cells are basically, so what happens is whenever these immune cells are maturating, so in that context, what happens? These macrophages and dendritic cells present antigens to these maturing immune cells. Okay, these antigens can be either self antigens or antigens from different pathogens or foreign antigens, what we can say. Fine. So these self antigens are basically antigens from our own body. Okay. So all these proteins, any other thing that is present in our body is presented as a self antigens to these immune cells. Okay. So that these immune cells can recognize what is a self antigen that is present in your own body and what is a foreign antigen. Okay, so if the cells that are against these self antigens are called as the self reactive T cells or the self reactive immune cells. Fine. If the number of these self reactive immune cells increases in the body, that can lead to any autoimmune disorder because we are directly targeting our own cells. Okay, is the explanation fine? Okay, any more doubts? Okay, so let's move further. So now let's move on to the secondary lymphoid organs. That is, so these lymphoid organs help in the further maturation, differentiation of the immune cells. Okay, differentiation and activation. So now we have different secondary lymphoid organs like spleen, lymph node, then we have MALT, mucosal associated lymphoid tissue, GALT, gut associated lymphoid tissue, and CALT, cutaneous associated lymphoid tissue. Okay, so these are the secondary lymphoid organs where differentiation and activation of the immune cells or the lymphocytes occur. Okay, so now first we'll start with spleen. So spleen is basically a major secondary lymphoid organ. Okay, and it is a largest secondary lymphoid organ where there is basically the trapping of the antigen takes place. Okay, and the production of antibody takes place in spleen. Okay, so this is the major role of the spleen. Talking about its structure, it has two kind of structure. One is the red pulp and the other one is the white pulp. Okay, so because spleen is also a graveyard of the RBCs, right? So it clears the blood from any kind of foreign particle or the waste material. Okay, so the red pulp basically helps in clearing of the blood. So that's why it is called as the graveyard of RBC. Okay, that's why it helps in filtering the blood. Now, the second component, that is the white pulp. So white pulp have different like regions or the different components. Okay, so first one is the primary follicle. This primary follicle is contains the immature B cells. Okay, that do not have interacted with the antigen. Okay, and these are the resting B cells basically. Then we have the marginal zone that contains the lymphocytes and the macrophages. So macrophages are the antigen presenting cells that engulf the pathogen and present it to different immune cells. Okay, so macrophages and the dendritic cells are the major cells that help in actually activating and differentiating different immune cells. Okay, so this marginal zone contains the lymphocytes and the macrophages. Then we have the periarticular lymphoid sheath that is called as PALS. So this PALS, this is rich in T cells or the T lymphocytes. Okay, and help in their activation and differentiation. Okay, so this then after their maturation they form when they have interacted with any of the macrophages, they come in the germinal center. So this germinal center will have activated or differentiated T cells, differentiated immune cells. Okay. Hmm. Then after their maturation, what will happen? After their differentiation, they are actually released into the blood flow, and then they reach the at the site of the infection or where it is. Okay, then what happens is, if there is any splenectomy, okay, the splenectomy in any of the individual will lead to, because it will not be able to trap any of the antigens or the foreign pathogens, so it will not be able to develop any immune response against that. So splenectomy will lead to any number of infections that can be caused due to the foreign pathogens. Okay, so this is the case with the spleen. So any doubts? One more thing, because these are actually the organs in which production of antibodies takes place. So these are the organs that are rich in the plasma cells. So plasma cells are those B cells that secrete antibody. Okay, and in spleens there is around 50% of the B cells that are matured and activated, while 30 to 40% T cells are matured. Okay, so it is a major site for the maturation and activation of the B cells rather than the T cells. Okay, so now let's move on to the other organ, that is the lymphatic system. So lymphatic system is an most important part of our body. It basically separates the blood vascular or the cardiovascular system from the immune system. Okay, and it is a sub part of the vascular system itself. So what happens is, when the blood actually flows from different tissues to the particular specific locations, it flows from the arteries to the tissue fluid or the capillaries, and then to the vein. Okay, so this tissue fluid from the capillaries is separate, and it leads to the different corners of our body. Okay, so these capillaries now filter out the blood into different lymphatic vessels. Okay, so these lymphatic vessels, that is also called as a tissue fluid, will carry lymph, and this lymph will travel into different lymphoid organs as well as the lymphatic system. Okay, so this lymphatic system basically is carries the plasma from the blood that seeps into the tissue. Okay, that is the interstitial fluid that either goes back or becomes lymph. Okay, so these actually this tissue fluid or the interstitial fluid actually finally becomes the lymph, and then lymph enters the lymphatic vesicles or the lymphatic vessels. Okay, so this is how this in the picture, as we can see here, this is a tissue space, and these are the lymphatic capillaries that actually filter out the interstitial or the tissue fluid. Then from this, they they actually travel to the different lymph nodes. Okay, so this is a efferent lymphatic vesicle, and this is the efferent. So efferent actually takes the lymph to the lymph node, and then the efferent one takes the lymph away from the lymph node. Okay, so now let's move on to the major or secondary lymphoid organ, that is the lymph node. Okay, so lymph node is basically a encapsulated bean shaped structure like this, as you can see. So capsule is will be present in all the lymphoid organs, as we have seen in thymus, then we have seen in spleen also, then here also in the lymph node. So a capsule will be present in all the lymphoid organs. Then it has a network of stromal cells that are packed with lymphocyte, macrophages, and the dendritic cells. Okay, then these are connected to both blood vessels and the lymphatic vessels. So these help in actually carrying all the blood related pathogens and the immune cells to different parts of the body. Okay, so briefly speaking, as the from the like in short, they have the efferent lymphatic vesicles. Okay, that like take lymph to the lymph node. Then they have efferent lymphatic vesicles that take lymph away from the lymph node. There are three regions in a lymph node. One is the cortex. Okay, the other is the paracortex and the medulla. So the cortex is the outermost region, then comes the paracortex, and then comes the innermost region or the medulla. Okay, so the cortex contains primary lymphoid organs, primary lymphoid follicles. Okay, as you can see here, this is a detailed description of the lymph nodes. Now, this cortex contains the primary lymphoid follicle. So, this primary lymphoid follicle is so this primary lymphoid follicle basically contains the B cells. Okay, so that have the immature B cells in it that do not have been interacted with any of the antigens. Fine. From this primary lymphoid vesicles, when the B cells are actually being matured, after maturation, they turn into the secondary lymphoid follicles. Okay, so these secondary lymphoid follicles have a characteristic center called as the germinal centers. So these germinal centers are rich in activated B cells. Okay, so these structures, as you can see here, are the cells that have the matured and activated B cells. Okay, then this was a part of the cortex region. Then in the inner side we have the paracortex region. So these paracortex region have basically the T cells and the dendritic cells in it. Okay, so where the interaction actually takes place. And then comes the innermost region, that is the medulla, that contains the plasma cells or the antibody secreting cells. Fine. Now there are certain more components. First is the post capillary venule that allow the migration or the circulation into the lymph node. Okay. So from these secondary lymphoid follicle, if the cells transfer to the medulla, okay, how can they transfer? They transfer through this post capillary venule that helps in like moving of the immune cells across the lymph node. Okay, then we have the efferent lymphatic vessel, as we can as I have told before also, this helps in taking the lymph away from the lymph node. So all the activated immune cells travel to the body through the efferent lymphatic vesicle. Okay, so this was it for the lymph node. If you guys have any doubt, you can ask. Okay, if you guys have do not have any doubt, let's move forward. So now we are moving on to the different questions. So this is the first question. What conditions highlights the consequences of the lymphatic blockage due to a parasitic infection? So what can be the correct answer? So the correct answer is yes, 3, that is elephantiasis. So what happens in elephantiasis is the lymphatic vessels. Okay, so the main lymphatic vessels of the body are affected because of the infection from a worm. Okay, so from because of the infection from worms, of these lymphatic vesicles actually get blocked. Due to lymph accumulation, there is swelling in different parts of the body. For example, this is the arms, then the legs, then we have the breast, the scrotum, the vulva that is in the mouth, and this is the neck and the face. Okay, so because of the infection of the worm, for example, we have Wucheria bronchitis. Okay, so these worms actually block the lymphatic vesicles that lead to the blockage in the lymph. Okay, so blockage leads to accumulation of lymph and leads to the swelling. That's why this is called as a, because this actually has inflated this much that it looks like the hand of an elephant. Okay, so that's why the disease is called as elephantiasis. Okay, so any doubts in this particular question? Okay, so let's move on to the next question. Which of the following statements is correct? So what can be the correct answer? Okay, any more answers? Okay, any more answers? Okay, so let's see the options first. Splenectomy can result in the systemic infection caused by Neisseria meningitis. Yes, this is correct. As I have told just now, whenever splenectomy or the removal of spleen takes place, so this results in infection due to Mistake in point C. Okay, I'll check. So, due to the removal of the spleen, different pathogens can actually infect the body. Okay, so major pathogens, for example, Neisseria meningitis, Haemophilus influenza, Streptococcus pneumonia, that cause major diseases, can actually infect the human body, and it will not be able to fight back because of the spleen is not there. Okay, so this is correct. The bursa of fabricus is not a lymphoid organ in humans but in chicken. So yes, this is also correct. The bursa fabricus is a major lymphoid organ in chickens where it helps in B cell maturation. Okay, so B cell maturation in humans takes place in bone marrow. So this one is correct. The size of the thymus is higher in children as compared to adults. So this would be adults. Okay. Mm, yes. So this option is also correct because the thymus size decreases as we go from children to adult. Okay, so from children to adult, when this thymus is actually, the size of the thymus decreases because the number of infections that takes place in the particular organs, so it that's why its size decreases. Okay, so this option is also correct. The appendix is a major site of B cell maturation in rabbits. Yes, this is also correct. As we discussed just now, the B cell maturation in rabbits takes place in appendix. With the advancement in age, bone marrow is mainly filled with fat. Yes, this is also correct. So, because what happens is the spongy or the red marrow basically decreases with age. Okay, then interstitial fluid either go back or become lymph. Yes, this is also correct. So, just now we've seen in the lymphatic system, the interstitial fluid basically have two options here. Okay, when it separates from the blood in the capillaries, either it go back to the capillaries or it become lymph and enter the lymphatic system. Okay, then lymph only moves in one direction. Yes, this is also correct because the lymph that is separated from the capillaries first would contain the pathogens, the debris, or the any of the waste material. Okay, then when it moves through the lymph node, then it would actually drop all the pathogens there, and from there it will carry the immune cells, the antibodies to the different organs. Okay, so that's why there will be only a single flow. Okay, so it moves only in one direction. Okay, so all of the options are correct. So the option 3, so this was wrong. So all of these statements are correct. So option 3 is correct. Okay, so any doubts in this particular question? Okay, so let's move on to the next question. What is the significance of using irradiated mice in immune research? So, what can be the correct answer? Okay, second, any more answers? Okay, okay. So the correct answer is yes, option number two, that is to eliminate their immune capacity, allowing for the testing of new immune therapies. Okay, so basically, what we do is we take a mice, so consider this as a mice, then we actually irradiate using either x-rays or gamma rays, and generally this irradiation is whole body. Okay, so that all the body hematopoietic stem cells dies. Okay, so that kills the dividing hematopoietic stem cells so that it will not be able to produce the new or fresh immune cells. Okay, so this basically is a like a part of the immune suppression. So if the dividing hematopoietic stem cells dies, this will be immunosuppression. Okay, now if a mice doesn't have any dividing hematopoietic stem cells, so if we actually test different immunotherapies, or okay, we test different immunotherapies or any g- graft rejection studies, organ transplant studies, and the studies related to cancer drug production. Okay, so these therapies that are related to the immune system itself would be actually helpful in these particular mice. Okay, so that's why these mice are irradiated so that their immune capacity is eliminated. Okay, so the option number two is correct. So, any doubts in this particular question? Okay, if not, then we'll move on to the next question. What process ensures that only self-reactive, non-self-reactive T cells survive during development in the thymus? So, what can be the correct answer? Okay, any more answers? Any more answers? One. Okay. Okay. So the correct answer is option number one. So let's see the options first. The apoptosis of T cells that react to self antigens, then negative selection of B cells, production of cytokines of by T cells in the thymus, receptor editing the T cell receptors, phagocytosis of the non-functional T cell by the macrophages. Okay, so what happens is, like I have said before, so first we'll understand what is self-reactive. Okay, so during the process of maturation, what happens when this T cell is basically presented by different antigens, by the antigen presenting cells basically. So these APCs, they present different antigens to the T cells. Now, these antigens can be either self antigens or non self antigens. Okay, self antigens means the antigens of our own body. Okay, and non self antigens are those that are foreign to the body. Okay, so both of these antigens are presented to the T cells. Now, these T cells will be now two types. First is that will be self reactive, that means against these self antigens, and the second one would be non self reactive against the foreign antigens. These self reactive antigens are basically used to by the immune system to differentiate between the self and the non self antigens. Okay, if these particular self reactive T cells become more in number as compared to the non self T cells, these would lead to autoimmune disorders. Okay, because then these self reactive T cells would directly be like acting against our body to degrade that particular antigen. Okay, so for to actually remove that chance, the thymus itself have a thymic selection. So this thymic selection basically helps in removing the self-reactive T cells from the body by apoptosis. Okay, so they generate certain signals so that these self-reactive T cells would actually go into the negative selection and it dies by apoptosis. Okay, here is another explanation. So there is a negative selection. So this is called as a negative selection of the T cells. Okay, and these T cell basically dies by the apoptotic pathway because these are the having major strong binding to the self antigens. But there are other T cells that are also self reactive, but they have weak binding or the intermediate binding to the self antigens. Those are retained in the body so that they can differentiate. uh, So they move to the positive selection so that they can differentiate and regulate the immune responses of the cell. Okay, so they also play an important role in the immune tolerance or the peripheral tolerance in the body. Okay, so any doubts in this particular question? Okay, if not, then we'll move on to the next question. Which of the following are the functions of the lymphatic system? So, what can be the correct answer? 3 and 7. Okay, any more answers? Okay, so the correct answer is option 1, 2, 4, and 6. Okay, so let's see. First is reclaiming excess interstitial fluid to maintain fluid homeostasis. Yes, this is correct. Okay, so what happens is whenever there is some excess fluid in the blood, so they actually reclaim the excess interstitial fluid and they move to the lymph. Okay, where it actually goes to the graveyard or the like sorry, yes, so they move to the graveyard. Okay, so now if the lymph is there, then they actually move to the recycle part. Okay, then producing red blood cells in response to low oxygen levels. Yes, this is also correct. When there is low oxygen levels, then it helps in moving the red blood cells and the production to compensate the low oxygen levels. Then absorbing dietary fats from the intestine. No, this is not the function of the lymphatic system. Okay, removing carbon dioxide from the bloodstream. Yes, this is correct. So all the waste products that actually is produced from the cells are moved by the lymphatic system. Okay. Then preventing tissue swelling through drainage of excess fluid. No, this is also not correct. Then producing hormones to regulate metabolism. Yes, this is correct. And synthesizing antibodies directly in lymph node. No, this is this can be the correct answer. Okay, so seventh one is also a correct answer because it helps in antibody synthesis in the lymph node. Okay, so here we can add the seventh one. Okay, So any doubts in this particular question? Okay, if not, then we'll move on to the next question. Mucosal surfaces play a vital role. Yeah, dietary fats are not absorbed by the lymphatic system. Okay, they are first absorbed in the bloodstream, then it will go to the then it will go to the accumulation part. But it is not it is not done by the lymphatic system itself. Okay, so mucosal surfaces play a vital role in immunity due to their unique structural and functional characteristics. Which of the following best explains their immunological significance? So, what can be the correct answer? Okay, any more answers? Okay, any more answers? Okay, so the correct answer is option one. So they are enriched with specialized immune cells and secretory mechanisms, forming a critical barrier to pathogen entry across extensive internal and extensive surfaces. Okay, so this is a basic structure of a mucosal associated lymphoid tissue. Okay, so this is a tissue that have this mucous membrane, and this is villi basically. Okay, so this villi actually helps in absorbing the particular pathogens from the mucosal membrane. Okay, so it also has, this is a epithelial cell, so it has a specialized cell called as a M cell. So this M cell has a porous structure that actually can engulf this antigen or a pathogen that is present in the near the mucosal membrane, and it will direct it to its pocket. This pocket have different types of immune cells. For example, it has B cells, it has macrophages, and it has T helper cells. Okay, so this macrophages and B cells will directly engulf this particular antigen and will help to initiate a immune response against it. Okay, so this is why this has a very important role in the body, and it is also a type of a secondary lymphoid organ, because here different cells actually interact directly with the pathogen here. Okay, and these M cells are a specialized structure that is present in these particular organ. Okay, so any doubts in this particular question? Okay, so let's move on to this one. So which of the following statements best describe the role of M cells in the immune system? So what can be the correct answer? Okay. Any more answers? Okay. So the correct answer is option number 3. That means M cells facilitate the transcytosis of antigens and pathogens across the epithelial barrier to immune cells in Peyer's patches. Okay. So Peyer's patches is another kind of lymphoid organ that is part of the gut. Okay, gut associated lymphoid tissue. Okay, and M cells, as we have seen in the previous figure also, so these are the M cells that are specialized cell to perform the transcytosis. Okay, so now what is actually a transcytosis? Okay, so transcytosis means that these particular M cells can actually take up the microorganism or the antigen from the outside, and it can transport this particular like pathogen or an antigen through itself to the other side. Okay, so this scenario takes place very rarely inside the body, where the cell can actually transport the component from one part to the other through its whole uh, cellular region. Okay, so this is a M cell. As we can see here, it would have receptors for the particular pathogen, and it can actually attract these in the gut lumen. Okay, and it transport it to the other side where different other cells are present. Okay, so this process of transferring this from one side to the other side is called as transcytosis. Okay, and on the other side, there would be different immune cells that would be present, and that would actually interact with this particular pathogen that have been transported by the M cells. So, M cells are a specialized cell that show the process of transcytosis. Okay, so any as and there is a specific receptor called as GP2. This GP2 is present in the M cells only. Okay, so as if in one of the publications, it is shown that the GP2, because it was marked green. Okay, So this is a M cell, as you can see here. There are various GP2 that are present across this particular M cell. That means it actually goes to the inside and it can transport to the other side of the cell. Okay, that's why this gap GP2 is actually intracellularly located also. Okay, so any doubts in this particular question? Okay, so let's move on to the next question. Yes. So which of the following cells is responsible for antigen phagocytosis in the skin? So what can be the correct answer? Okay. Any more answers?
Sorry, sir, joined late. Can you just briefly tell me how to answer
Sorry,
this
sir,
question?
joined late. Can you just
Should I
briefly
write in
tell
the
me how
textbook
to
or
answer
how
this
is it?
question?
You can write in the
Should I
textbook.
write in the textbook or how is it?
Okay. So the correct answer is option number two. That is the Langerhans cells are the primarily phagocytotic cells that are present in the skin. Okay, and here is the explanation. So the M cells are basically the cells that can engulf the pathogen, but it cannot degrade it. Okay, so degradation will not happen in the M cells. It can happen in those cells that can phagocytose the pathogen as well as degrade it. That is done by the Langerhans cells. Okay, so the Langerhans cells are basically the immune cells or the tissue specific. Okay, tissue specific macrophages. So these are the tissue specific macrophages that are present in the skin and they'll help in actually phagocytosing the particular pathogens that are present on the skin. Okay, as we can see here, this is a mouse skin and this is a human skin. And this epithelium is more in the human skin as compared to the mouse skin. And these Langerhans cells are present in this epithelium. So these are tissue associated macrophages present in the epithelium of the skin cells. Okay, So any doubts in this particular question? Okay, if not, then we'll move on to the next question. Just 1 minute. No, these are actually the questions that have been asked in the last year's the last year's exam. Okay, so we are actually being discussing that based on the concepts that have been taught in this particular week. And you, if you have missed any part of the discussion, these videos will be uploaded in YouTube as well, so you can see those videos in that particular uh, in YouTube also. That I don't know whether the final exam would be MCQ type, but we are trying to actually like clear out all the doubts and all the important questions that have been discussed previously. Okay, so let's move on to the next question. If a knockout gene affects embryo development in the animal models and results in lethality, which strategy for gene knockout could be useful? So, what can be the correct answer? Okay, any more answers? Any more answers? Okay. Okay, so the correct answer is option number four, that is a conditional knockout. Okay, so what is actually a conditional knockout? In basic terms, conditional knockout means if any gene actually cause lethality, that means in animal models it will not be able to produce any organism. Okay, so if no organism will be produced in that case, then no study will be performed. So, in that case, what we do is we generate. So, these are the conventional knockouts. That means whole body knockouts of that particular gene. Okay, so these are whole body knockouts. So, if that is the case, then we generate conditional knockouts. So conditional knockouts are the knockouts in which if we have to study, for example, brain, then that particular gene is knocked out in the brain only. Okay, it will not be knocked out in any other region of the body. So it will be knocked out in brain itself. And then we can study that, okay, this particular gene will not be expressed in brain. So what will be the effect if this gene is mutated? Okay, so that is the meaning of the conditional knockout and difference with the conventional knockout. Okay, and how do we develop conditional knockout strains? So there is a important mechanism that takes place, that is generated with the help of recombinant DNA technology, that is the Cre-Lox system. Okay, so Cre is a recombinase that actually recognizes the LOXP sites, and it cleaves the LOXP sites where it actually sees them. Okay, so this is the basic example of the Cre lock system. So Cre is a recombinase that recognizes these lock sites, and it cleaves anything and removes anything that is in between those lock sites. Okay, so this is the basic mechanism how a Cre recombinase works. So for example, this Y gene is between two LOXP sites, and this Cre recombinase actually works, and it actually takes out this Y gene and removes it from the body. So this Y gene is actually inactivated. Okay. Now the positioning of these LOXP sites actually helps in uh, producing a deletion, inversion, or a translocation. Okay. So if they are oriented in the same direction, that means it leads to deletion. Fine. If they are oriented towards each other, that means it leads to inversion. So if a gene is produced in this direction, if the log p sites are inverted, that means it will be now inverted in the other direction. Okay. And it helps in translocation by removing this particular thing into the other region, and it can actually, with the help of other enzymes, we can integrate the part removed from one part to the other region. Okay, so this is a basic mechanism how a Cre and a lock system works. Now, how we have actually exploited it for the conditional knockout system. So if there is a Y gene that needs to be knocked out in brain, for example, so we generate a mouse strain that have this LOXP site around this gene Y, and this is generated as a whole body thing. So wherever this gene is present, it will be present with the LOX sites. Okay, now we deliver a the Cre recombinase with a promoter that will express only in brain. So we have a tissue specific promoter here. Okay, so we have a tissue specific promoter here. So this promoter will only be expressed in brain, and when this will be expressed, that means the Cre recombinase will always all will be actually expressed in brain itself. Okay, it will not be able to express itself in liver or any other part of the body. So, if this Cree will be developed in brain, it will actually knock out or remove this particular Y gene that is present in the lock sites. Okay, that will lead to inactivation of gene Y in tissue X, that is brain. So this helps in producing conditional knockout mice. Okay, so any doubts in this particular mechanism? Okay. If not, then we'll move on to the next question. So which of the following statements regarding stem cells are correct? So what can be the correct answer? 1, 3, and 8. Okay. Okay. Any more answers? Okay, so let's start seeing the options. First is transgenic mice can be developed from pluripotent stem cells. So, okay, 1 and 8. Yes. So, the pluripotent stem cells are basically the ones that can give rise to any cell except the placental cells. Okay, so the transgenic mice are developed from the pluripotent stem cells itself. Okay, so yes, this is correct. The pluripotent stem cells can give rise to all cell types, including the placental cells. No, this is false. The pluripotent stem cells can give rise to all cell types except the placental cells. Okay, unipotent stem cells are restricted to differentiate into only one cell type. Yes, this is correct. So, the meaning of unipotent means that they are actually differentiated to a single cell lineage. Okay, so that means they can form any tissue that have a similar function. Okay, of that particular lineage. So this is also correct. Then we have the fourth one, embryonic stem cells are used to develop knockout mice. Yes, this is correct. The embryonic stem cells are basically the pluripotent stem cells. Okay, so yes, they can develop the knockout mice. So this is also correct. Embryonic stem cells are not used to develop transgenic mice. This is false because embryonic stem cells can be used to generate transgenic mice because they are pluripotent in nature. Multipotent stem cells can't generate biochemically related cell types. No, this is false. So, multipotent stem cells basically develop different cell types that have a same function. Okay, so similar function cells can be actually developed from the multipotent stem cells. Okay, so this is false. Then totipotent stem cells can't develop the placenta. No, this is also false. So totipotent stem cells are the ones that can generate any type of cells, including the placental cells. Okay, so this is also false. Then totipotent stem cells can form both the organism and the placenta. Yes, this is correct. So the correct answers are first, third, fourth, and eighth. Okay, so any doubts in this question? Okay, if not, then we'll move forward. Which of the following statements about lymph is true? So, what can be the correct answer? Okay, any more answers? Okay, any more answers? Okay, yes, the correct answer is option 2. That means lymph because it is separated from the interstitial fluid. Okay, it actually enters the lymphatic system as lymph, and it have all the antigens, the waste products that are removed from the blood. Okay, so yes, the correct option is answer number 2. Okay, so any doubts in this question? Okay, if not, then we'll move forward. So this was the last question for today. If you guys have any doubt regarding any discussion or the question, so you can ask. Okay, if no doubts, then we'll close the session for today. Thank you guys for joining in. We'll meet next week.